Hello, everyone. Today I'm gonna introduce my research topic, which is identifying the performance of Asian DNA sequencing methods. I am the main speaker. I'm the speaker here of this presentation. I'm Yuzhe Yan, and I'm glad to introduce my supervisor, who is Indu and Aidan. Uh, the first, I'm going to introduce some background information about our research. So first problem is, what is in-solution enrichment? The in-solution enrichment is technique used to isolate and amplify specific DNA sequences from complex mixtures, such as those found in the like Asian samples, especially for Asian human DNA samples. In-solution enrichment is particularly valuable in Asian DNA research. And second problem is, why abstract archaeological sample from Asian DNA is really important. Uh, so the extracting Asian DNA, especially for Asian human DNA, is allows to researchers to directly study the genetic material of past populations, offering insights into human evolution, migration patterns, and interaction between the Asian groups. So the genetic evidence can complement traditional archaeology methods, like by scientific methods, bioscience method combinations, providing a more comprehensive understanding of historical events and cultural developments. And third, like what are the important measurements? So I feel like measurements are the variables from our data I can introduce to you guys. The first one is SMP. SMP is a single nuclear polymorphisms, and SMPS is like several multiple. We have 15 here in our data, single nuclear polymer systems. And the BP means base pairs. And normally we're understanding like uh, the effective base pair is like 30 BP. That means the unit of the length for the DNA is around like 30 or over 30. And the next one, 1240K. 1240K is a collection of 1.24 million SNPs, the DNA sets on the genome. And as we know, its approximately number is not very quite because it's huge. And the next is coverage. The coverage is like means number of atoms in the heat of DNA, because we know DNA is combined by the GC right? So we can know probably it has one position heat, two position heat, or zero position to heat. And next is percent endogenous DNA. And the percent of our DNA, like the different DNA soup that we decide is a human or not by human. So this ratio is actually pretty important at our future analysis. And the next problem is, what makes DNA damage or contaminate? Because so our research targets is compare the two methods, which can help us to get like the good DNA, the partial fragments. So why makes the DNA damage? And DNA damage and contaminated by several issues. Like we know, like environmental or the chemical degradation can cause the DNA damage, like overheat environment or like extremely cold environment can case of that. And the chemical degradation, like because those bones basically are derived from those like Asian human bones. So like those bones contains by maybe like soil, rock, bacteria, or some different like variety environments. So we don't know like what. So those can cause like chemical degradations, like they affect each others. And the next is like contaminations. So DNA contaminations like normally affect a lot. Uh, so like 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 the no like I mean the contemporary social human DNA effect like contaminate the Asian human DNA. The ancient bacteria, ancient animal also like contaminate those DNA. So they mix together. So that's why I mentioned before why the endogenous ratio is important. And next, yeah. And the next part is objectives. Before we talk about our objective, we know who's our research target. As I mentioned before, there has two methods. The first is 1240K reckoning method. The 1240K reckoning is widely used for in-solution enrichment of Asian DNA and the twist Asian DNA assay. It's kind of a new method, and people all scientific I can see like commercially use them to design efficiency capture of Asian DNA sequences. But there's a little bit difference because the twisted method use double standard 80-based pair province and require only one round of enrichment. So logically, technically, it sounds like better than 1240K, but we'll figure it later. Talk about our main objective. As the topic shows, we need to compare the performance of them. So how to compare performance. So here it is. To evaluate and compare the effectiveness, efficiency, and overall contribution of the twist Asian DNA method and the 1240K, with a focus on improving our understanding of human evolution history and genetic diversities. The significance for the human evolution studies is actually, like by comparing those two methodologies. So our research, my research, our research will contribute to optimizing Asian DNA analysis protocols and in enabling more accurate reconstruction of human evolutionary histories. We know where we're from, what we got. Maybe we have the same part of compare our like the Asian humans. So the inside gene will be aid in understanding major demographic events, migration pattern, and genetic underpinnings of Asian human populations. So like in conclusion, the comparison study like for this project aim to identify the most effective and efficiency method for right now. Maybe I have some new method in the future, but right now we find a better one. So like advancing our knowledge of human history and evolutionaries. As you can see, like so this sentence is what I take from the one science essays. And the author is called Shao-Chang. Shao-Chang mentioned the outcomes of this research will not only enhance methodology standards in the field, but also can get knowing information about how people connected in the Asian world. It's also comparing several methods to analyze like Asian human DNA. All right, next part is our data. The data is very important because we're using those data to analyze to get our like target. As you can see, I use the scheme function. Scheme function can like to present summary about selected variables. We have 49 rows. That means we have like 49 subjects can do the analysis. And under the items constructions, so I and our like side group like 10 like useful variables and implementing to the data set, we have 15, 17 right now. You can see there has like S&P 1240K, input rates, macro rates, indulgence ratios, as I mentioned before, coverage ratio, GC content ratio. GC content is actually, it's a funny one. I will introduce it later, and content ratios, contaminants, contaminations. So, and I list under it. And next, we're going to talk about like methodologies. Our statistical or data science methodologies for analysis, the eager data. The last trimester, what we learned and what we used is linear regression and logistic regression. And you can see here, the figure 3 and figure 4. Figure 3, the linear regression for the S&P 1240 case and input rate. No matter from the jitter or like the line chart, we can see, smooth line chart, we can see the twist is way outperformance than the 1240K. And the right figure is logistic regressions for the endogenous DNA and input rates. You can see it's not like, so this logistic regression is not like academic what we learned before, like this like logistic regression, but it's curved, and it also used like the functions to shows their who's better. Because we compared about the ratios from 0 to 1, we can see like the twist has a better, higher ratio than the 1240 case. This one is the functions in summary about like additive GLM function, the logistic regressions. You can see it's not like significance from the p-value here, we can see, but since we only need to compare those two methods from the endogenous side, so we see it's pretty straightforward. And here is linear regression summaries. It also works. This one is, if we talk about like models, is better than the last one. It's very significant here, and we know their relationships, and we know who is better. And talk about the right side, this big screenshot. This is a new method we learned from this trimester. It's mixed effect model. So I set S&P 1240K and increase at fixed term. So But this is like actually automatic expectations because all the p-value is significant. That means I successfully like made this mixed effect model and to successfully gather relationships. And next, here's some results what we get. So after several comparisons, I figured out that the twist is really better than 12-week case. The answer from my side, is yes or no, because as the source comparator can see here, we can see the twist is majority best, better than 12K, but still have several parts. 12K is better than twist. But is that real? Is that really like 12K better than twist? So we talk about like GC component like ratio. So like GC is a proportion of nucleotide that are wandering or the critical signs has downgrade by. So it's connected with bias. The higher GC means higher bias. So we don't want like our analysis result had higher bias. So definitely the higher GC you can see 12K is bigger than twist, but this shows 12K. The method actually is not good at twist because it has connection with the bias. So that means I have to do more researches and maybe I have to implement some more variables into it to figure it out what's like something else areas probably can make 12K better than twist. And as you can see, it has the life cycle table too. It's from the Ronald essays at Ronald published at 1222. All the data is from the like official library, so it's like official tested, is being tested. Here is 12K coverage and two twist coverages. And we can see they also have 12K X component, Y component, 12K Y component. If we check this area, we can see still twist has a lead at majority of area, but not everything. So I made this line chart here to compare which area like they perform better. As we can see, pretty obvious the twist is better than 1240K, but still not all of them. You can see here, yeah. As I mentioned, the research is not finished, so I will dig in more infos to do the model research to maybe to do the model readings to figure it out and to test what I guess is correct or not. Talk about like future work, like it's not being finished, so you have like 6 or 7 weeks to finish it. So we got new data. So I will prepare to use new data to do some like deeper analysis. I filter genotype. So I'm prepared to using uniform distribution to analyze those 3 subset data set. And as I mentioned before, I'm planning to implement more variables. So I need to figure out which variables are really useful and which of them are not, are useless. So I will use PCA to take off or the dimension some useless variables in the future, and also the bias analysis. If it has a bias relationship with some variables, how should I deal with it? And I will also make more discuss with my classmates and also with my supervisor to make sure my personal thinking is close to right. It should be not off right, but at least to be close to right. Here's some reference about like the Asian human DNA researches, and also some statistical and data science methods. And thank you guys. Thank you guys to hear me. If you have any questions or the concerns or the related thinking, just email me or text me. I'm available at all the rest of the trimester. Thank you. And thanks for all my audience and my supervisor. Thank you.